I don't mind having saying a few dumb things while we're counting down, because I clip this out of the proper video, but I don't clip it out of the live stream that stays up on YouTube forever. So hello, live streamers. If you've stumbled your way onto thebirdemergency.com live and found this, those are beautiful penguins from the subantarctic, adalies. I think there's some gentoos in there too. There are some gentoos in there too. Gentoos with the orange feet, adalies with the pink feet. 10, no, 8. Hello, bird nerds. I'm Grant Williams. This is the Bird Emergency live, coming to you from the wonderful city of Naarm, and it is Reconciliation Week. So I would like to pay my respects to the traditional owners on this unceded land that we colonialists call Melbourne. Today, right out of my comfort zone, right out of my wheelhouse. So let's see how we go. I'm joined by Dr Stacey Preistley, who is based at the University of Adelaide, and she's a hydrologist. And what kind of chemist did we say?
Geochemist.
A geochemist. And we're going to talk about the work that Stacey's doing on Project Coorong. For those of you who don't know, the Coorong is this amazingly long, skinny waterway, shallow waterway as well, that's sort of at the mouth of the Murray, but not right at the mouth. But Stacey can tell us about why it's so important. But if you're as old as me, you're going to remember the Coorong as being the location where Storm Boy was shot, which was, for Australians of my vintage, that was our first introduction to Greg Rowe, who played the original Storm Boy, and we met Mr. Percival the Pelican. And for a lot of us, our first introduction to the wonderful actor David Gulpilil. I'd seen him in Jeddah as well before that, but let's not get too specific. Stacey, welcome to the Bird Emergency. Tell us what it's like working at the Coorong. I had to do it. Still got election fever on the brain at the Coorong. And tell us why the Coorong is so significant. Then we'll sort of explain where it fits in the whole Murray-Darling Basin scheme. Floor is yours.
Thank you. So the Coorong, well, firstly, it is just a beautiful spot with tons of bird life, hence why I guess we've got this connection. So it's a pretty famous bird watching spot. And it is located at the end of the Murray-Darling Basin. And the reason it is such a sort of iconic site is because it's got this really great gradient of freshwater turning into saline water. So it's got really diverse habitats all along the Coorong. And part of the reason for that is because it is so long and skinny, with just one really significant opening where freshwater comes in. And so you get this sort of saline gradient where it becomes more salty towards the end. So there's various different habitats all along it. And it's, just a really beautiful spot. However, like a lot of great places in the environment, it's not doing amazing at the moment. And so we, as part of Project Coorong, the Healthy Coorong Healthy Basin, which I'll explain a bit later, have been asked to look at some of the issues that are occurring there so that we can help eventually manage the system better, so it will continue to be a great site for lots of different birds and fish and everything else that live in there at the moment.
All right, let's sort of talk about what does live in the Coorong. Is it a significant habitat in terms of its national significance for perhaps any fish or crustacean, mollusk species apart from the birds?
Well, I mean, the main thing is it's a site where a lot of spawning happens. So a lot of the native fishes and even I think it's marine fishes do some spawning in there. So it's quite a big uh, site for, I guess, like the life cycle of some fish species. This is very much outside of my knowledge base, so I'm just going by what I've overheard my colleagues talking about. But yeah, so it's a really critical spot for a lot of like fish life cycles. But I think the main one is actually the bird life, why it's so famous, and that's actually why it's been listed as a Ramsar site as well. So it's a wetland of international significance because of the bird life that it supports through all these fishes and different aquatic plants and the shorelines and everything.
How would you categorise that long, skinny waterway, which is, uh, I think it's about 90 kilometres long or something, isn't it? So it's
Yeah, I mean, it depends what you read, but yeah, it's just over 100 kilometers if you literally go from like tip to tip. I think it's like up to 140 kilometers.
yeah,
I've read some places. It's extremely long and extremely skinny, which is part of the reason why it gets that saline gradient occurring, which also then causes those sort of like various habitats along it. So it's a
and
very interesting location.
now would you call it a lagoon?
Yeah. I mean, it's the Kuron Lagoon. So yeah,
Okay, so
yeah,
it is a lagoon, and
yeah.
just to give an idea about how shallow it is, and that's you doing some of your work, um,
Yes.
and and uh, look, it's many years since I was there, but I've driven and been bird watching along a large portion of it. Uh, I never tried to walk across it, but I think you can pretty much walk across, and it's only just sort of head height at its deepest point.
Well,
And
I think
and I
some
think
of the
that
spots,
fluctuates, doesn't it, year from year,
yeah. So
depending
some of
on
the
water flow.
spots that can be, I think it's up to 2 meters depth, but for the most part, it's about a meter deep, and especially in the center of the lagoon, there's a location called Hell's Gate or Parker Point, and that is very shallow, and that's um, part of the reason why the South Lagoon. So I should explain that Parker Point separates the north part of the lagoon from the south part of the lagoon. Um, and at that point, it's very shallow. And I think you could probably easily walk across. In fact, it's very thin, like very narrow as well there. So you
Well,
could definitely walk across at that point, I think.
yeah, I I think that must have been. I must have gone there when I was a kid because I did walk across the lagoon at one point when I was about 8 or 9 with
Yeah.
when I first went to the Coorong. Um, to make it easy for people, Stacey, when I've got around to doing the editing and everything, I'll insert a map and some pictures
Yeah,
of
thanks.
these places that we talk about,
Yeah,
uh,
no, that would be helpful.
which does make it easier. And I'll also put a bit of a summary about the bird species that are regularly very common there, and some of the oddities and rarities that have been seen there. I don't want to go off onto bird stuff because that's not.
Yeah, I was trying to quickly read up on some of
Yeah,
the bird species that are
no.
just in case.
No. What I'm interested in is uh, what do we know about the health of the Coorong in, let's put it in sort of maybe a 25-year time period? Is it in the last 25 years, has it been getting significantly less healthy? And what defines health when we're talking from the point of view of a hydrologist and a chemist?
Yeah. Well, I guess just we still define health in terms of the ecology. So I guess as a geochemist, that is because I'm using chemical elements that are naturally abundant in the system to look at it, but we're still doing that for the purpose of protecting the ecology that's there. So to go back to the first part of your question, so the Coorong health has been declining over time. And so the part of the reason for that is because the majority of the fresh water that goes into the Coorong is sourced from the Murray-Darling Basin. And so as that's been progressively used for agriculture and the flows have reduced, we've been having less fresh water like flushing into the Coorong. And that has caused a buildup of salinity and nutrients in the Coorong. And so it sounds ridiculous in some ways that high nutrients is a problem, but it's like anything, like too much of a good thing is bad. And so with high nutrients, you actually get really unhealthy ecological processes happening, like runaway algal growth, and that's what's been occurring in the South Lagoon. And that's been particularly pronounced after the Millennium Drought. And so that was where the health of the Coorong was like significantly degraded. And it's because of that the state and federal government did implement Project Coorong, and since Healthy Coorong, Healthy Basin the ultimate goal is to improve the health of the Coorong, and where we come into that is because they need to know how it's working. So our research is looking at what's currently happening with the health of the Coorong and the nutrient cycling so that they can ultimately manage it better so that it becomes a healthy system again.
Now, let me ask you to define what you would think is healthy and what are the kinds of indicators that are used for that. Because if we're talking ecology, are we talking the number of species that are present, the size of those populations, the diversity of those populations, things like reproductive success, or are there indicators like salinity, turbidity, or any other things like, I mean, we haven't even started with isotopes, but
no, this is all good.
So because this is something that frustrates me all the time when we have all these projects, and there's a lot of publicity about them, and you just said the government's aiming to get it back to health. I want to know what the target is so that we can know if we have achieved our aim. Healthier is not healthy. That's my...
No, and they do have set targets. I could not tell you the numbers, but I can give you the general gist. So pretty much what you were explaining is exactly what we're looking at. Like there's salinity targets. So most of what I'm talking about is focused on the South Lagoon because that's the area of the Coorong that's the most degraded, and that's what the ultimate aim of sort of like fixing is, so that if the southern end of the Coorong's fixed, then that means the northern lagoon's still going to maintain its health as well. So one of the major issues at the moment in the South Lagoon is there's these, they're called monosulfidic black sulfide deposits. So they're these really disgusting black.
Is that the stinky black sludge?
That's the stinky. Okay. So it smells like rotten eggs. That really sulfidic type smell.
And we've all been to a fishing spot or something like that where we've just gone, oh my God. And it's just a graveyard for life, isn't it?
Yes. So, and that's part of so that's one of the sort of key indicators that it's really easy to see as well. Like if you walk along the South Lagoon, even along the shoreline in some spots, you can smell that smell and see that black mud. And so that's a really clear indicator that it's not healthy. So the fact that those muds are being deposited is because like healthy nutrient cycling and plant growth is not occurring, and the sediment itself is becoming anoxic. So there's no oxygen in the sediment. So these muds are what occur with that anoxic degradation of like a build up of organic matter. So that's one of the key things that we're trying to eventually get rid of, I guess, so that you can have then like plants growing there once the sediment is healthy. Because at the moment, those disgusting black muds are part of the reason that the plants that we want to grow there can't. The other thing is the salinity. Like a lot of these plants and animals have a maximum salinity threshold where they can survive. So that's one of the major indicators that the government will be using to sort of manage the Coorong, will be to keep the salinity below a certain concentration. And to do that, you need to get freshwater flushing into the system. And that in turn helps with keeping the nutrients down, because the water that comes in has got lower nutrients than the water that's already there, which helps remove some of the nutrients. So I can't say what the exact values would be, but keeping salinity down, keeping nutrients down, and getting rid of the black ooze sediment is sort of like the three key aims. And that's what the government will be like making sure they're achieving to manage the system.
Okay, so water flow, the inflow is one issue. Now, it's a very, very long, skinny lagoon, which we've just talked about, and that it's fed at the north end by the Murray River, which is
and the ocean,
and because of the mixing in Lake Alexandrina. That's
well,
right.
the water comes in. So the Murray-Darling Basin kind of discharges into Lake Alexandrina, which then like discharges into the Coorong. So the ocean water and Murray River water mixing is more in that Murray Mouth zone, and sort of as it's coming in to the Coorong. But yeah, so this is where with the Murray-Darling Basin Plan and the environmental flows water has actually helped already. So that's helping to discharge sort of like big pulses of water, which then can actually get into the Coorong. Whereas like before, I guess, especially during the Millennium Drought, when there was just trickles or no water, then it's just ocean water that's coming in, and you need those big pulses to really get down to the southern end of the Coorong.
And I think, correct me if I'm wrong, but my understanding is that over many cycles, that the mouth for Lake Alexandrina sometimes will close, and that's natural, but most of the time the flows through the Murray-Darling system keep it open in some way, which means that some salt water from the ocean is coming in, but we've degraded the water in the system, in the Murray-Darling system, to the point that so much of that water is now what we would call saline rather than fresh, even though it's not saline to the same way as the ocean.
I mean,
is that
no?
True?
So the River Murray water that comes in, I'm just trying to think of the salinities, but they're still very, very low. So when the River Murray water is there and it flushes in, it does have relatively low salinity and quite low nutrient concentration. So the water coming from the Murray-Darling Basin, when it does flow into the Coorong, is like replenishing the system with fresh water. So and there's always been some
so it's
mixing
not
of ocean water
saline.
there. No. So the River Murray water that comes in is good. It's helping the system freshen.
Using that measure, the salinity, the water quality is significantly better than it was. Okay, so that's working.
The issue is probably, I guess, the amount of water rather than the quality of the water coming
So that
from
was the next thing I was going to get to about the health of the water.
Yeah. So the water health is good. It's the quantity, but and that's where the environmental flows have helped, because they're providing more of that water to flush into the Coorong.
Okay, so the concentration of nutrients, which is what you are specifically interested in, is that, where are those nutrients coming from? Is that upstream in the agricultural uses and returns of water runoff in the Murray-Darling system, or is it let's talk about where they come from,
and then really important question.
And so where do they come from, and then
the big things we were looking to answer. So, I mean, there has been changes in, I guess, the land use around the Coorong and obviously along the Murray-Darling River, which might have added some nutrients to the system. However, that's not the cause of the buildup of nutrients in the Coorong. So the issue with the Coorong isn't that more nutrients are coming in, it's that there's less water coming in that's flushing out the excess nutrients. So it's not about too many nutrients coming in, it's about not enough removal of nutrients.
Okay. Now, before we follow up on isotopes and all that stuff, because that's where we're going to go, but if we had historically normal, and by historically I mean going back hundreds of years, not 20, 30, 50 years, which is how we tend to think of historical in our society, but that's not how it has normally been. How would the Coorong have normally flushed in times of massive floods and unimpeded flows down the Murray and the Darling and the whole catchment? Would the Coorong have overflowed and broken the banks back into the ocean to actually drain out, or would it have all had to cycle back up into the mouth, the very north end of the Coorong? What would have happened in days of yore?
Yeah. So I guess if you're looking back, like pre-European settlement, there would have been no gauging of the river or anything. So it would have been, I guess,
they would have been massive
more
floods,
like pulses of water coming through. So rather than the trickle, and it's those big sort of like flushing events that like really push the water into the Coorong, and like the really big events can get like the fresh water from the Murray like all the way down into the Southern Lagoon. And that's those big flushing events that occur. But I don't think there was any evidence of the Coorong being flooded to the point where it like breaks its banks or anything. Okay, it's just these like flooding events that really push in that fresh water. And then obviously if you don't have those flows, evaporation of the water towards the southern end especially just causes concentration of the salts and nutrients that are there. And then you need that big pulse again to sort of like push in fresh water and then push out some of that old like salty water.
And the point of asking that the way I did was that if we had that volume of water still coming in and getting into the South Lagoon, what you would then have is the natural biological environmental processes would be healthy enough to deal with the nutrients that come down, and they'd then be cycled in the natural way and wouldn't build up to be the sludgy, stinky, nasty mess that we get now.
Yeah, so the Millennium Drought really sort of like didn't help the system and really but I think really made these problems a lot worse, a lot quicker, if that makes sense. So we were potentially not seeing enough flushing, but there was enough buffering in the system to cope with that. And then the Millennium Drought happened and it just sort of made these problems like 20 times worse because there was like literally no water coming in from the Murray at some points. So since then we have had some improvements. and some of the work we've looked at was looking at some of these flushings that have occurred since the Millennium Drought. And there's been some particularly high flooding events. I think off the top of my head, in 2016 was one of them. And we did see evidence of some good flushing occurring then. It's not been enough to undo the damage that's already there. So you need to sort of rehabilitate the system, and then hopefully these flushing events, once managed properly, will maintain health. But they're not enough at the moment to like rehabilitate the system completely, which is why we need to sort of look at other things.
Well, we'll come back to that whole idea of rehabilitation and everything after we've.
Yes.
Let's trace the story of nutrients. Like how do you trace nutrients? How do you work out where they've come from? And I'm guessing you're also able to work out how long they've been there. So tell us what you do.
Yeah, so this is where isotopes are magic. So just in case people don't know what an isotope is, so an isotope is an element has different isotopes, and this just means it has different number of neutrons in its cell.
Electrons and neutrons.
Yeah. So the protons are the number of protons sort of determine what the element is, and the number of neutrons can kind of change without changing what the element is, but it changes its physical weight very, very subtly. So it does behave slightly different in the environment. So chemically it reacts the same, but it has, with that different mass, it physically behaves a little bit different. And so those really subtle changes in behavior in the environment are what we then look at and use to piece together what's happening in the system. So it's a bit corny, but I think of what I do is like CSI kind of, but with water and isotopes. So we're taking all the different like pieces of evidence that we get with looking at like nutrient concentrations and the isotopic ratios of various elements in these nutrients to figure out what's actually occurring, like where they're coming from and how they're cycling, so that we can understand the system a bit better. And you unfortunately can't do that just by looking at concentrations, because you can't tell from the concentration where the majority are coming from necessarily without looking at the isotopes. You need that extra piece of information to really, really kind of unpack what's going on at a really detailed level.
Now, the photo that we're looking at now, which is you standing waist deep, with handing back an apparatus that's connected by a long cable to some reading device to your colleague. Now, you're taking a measurement of something like salinity there, aren't you? Where an electrical pulse. That's an electrical pulse of some description. So you're not taking a water sample in that photo.
No. So whenever we do take a water sample, we pretty much always use this apparatus, which is called a YSI, to measure some of the properties like temperature, electrical conductivity, which is the salinity, the pH of the water, the dissolved oxygen concentration. So that they're some of the like baseline information we need to help us interpret the other sort of more detailed information or analyses we get from taking water samples.
So the methodology would be that every time you're on site, you would take those readings. They're your baseline.
They're my baseline. Yep. Even if I'm not doing much, I might just take a reading for why not?
Yeah, that's right.
Exactly. If I'm just waiting on the bank for somebody else to do something, just take a reading.
But when you take a water sample, an actual physical bit of water in a jar or something, I'm guessing you're probably doing turbidity, which is a pretty simple one that we did at school. But then you're doing the analysis for your isotopes of different elements. How are you doing that? Is that mass spectrometer? What kind of tool do you use? And how hard is it? How long does it take?
Yeah. Well, I mean, it depends what you're doing. So I mean, the water sample you can take the water sample and then filter out all of the like larger organic material. So you remove all the algae and everything out of that water sample to leave you with the actual nutrients that are dissolved in the water. And alternatively, you can like look at more of the larger organic materials like the algae and something we call particulate organics. So we can do a lot of different things on site with what we're taking to target specific things to look at. So yeah.
I'm sure most people don't have any idea how it's done. So the initial filtration, how does that get done? And then, like, is it like we see on all the forensic lab shows? Does it get put in a machine and get spun around, and then you pick it up, and there's all the different layers? Or take us through the day in the life of a water sample.
Okay, so but one of the things that we were trying to do was we wanted, as I just said, we wanted to get the dissolved nutrient species. So for that particular type of water sample, we would take some of the water into a syringe, so it's a bit bigger than the normal,
a big horse needle
injection syringes, like a bit bigger, and you have they're just like filter papers, but they come in little cartridges that fit to the syringe, and so they have like various different pore space sizes. So depending on how big or small
what you're sampling for,
you want to
get
trap
the right piece of paper, and then you force it through, and then you're left with what you want.
Well, yeah. So either way, you're either removing what you don't want from the water.
Yeah,
so
and then that is
dissolved nutrients. Then you would push that through the filter paper, and so then at the end of
it, that's what you want.
You've got the water sample that's had all of the algae and the big particulate organic material removed. So you're just looking at the dissolved
solid
nutrients. Alternatively, we also did want to look at what the isotope signatures are, or some of those larger organic materials not, I guess, contaminated with the dissolved nutrients. And in that case, we literally had, a piece of filter paper, and we passed water through that a set volume, and then we collected that filter paper, and that's then what went to the lab. So with that, we were looking at the algae and the larger particulate organic matter. And so it's those different. So for one of the analyses, you're looking at a filter paper with sort of the material on it, and then another analysis, you're looking at the actual water sample itself and trying to get the nutrients out of that.
So when you've got that, so I understand. I hope everyone does that. In one of those things, what passes through the filter paper is what you want to look at, and that's materials that have been dissolved and that are small enough to get through the filter paper and that you want to analyse. The other bit is the stuff that's too big to get through the filter paper is they could be dissolved materials or they could be suspended, but they're taken out and you would then analyse the bigger bits. But you're still analysing for the same type of things, aren't you?
Yeah. So one of the main things that we looked at, like with tracing the nutrients, was looking at the nitrogen isotopes. So nitrogen is obviously one of the main nutrients. So that nitrogen, phosphorus, and carbon are sort of like your big main nutrients in these systems. So we were predominantly focusing on the nitrogen system. So we were looking at nitrogen isotopes. So we'd look at the nitrogen isotope signal of the dissolved material in the water sample, as well as what we collected on that sort of the filter paper, which we call the particulate organic matter.
Now,
And
is
so,
is the nitrogen that's in the dissolved sample a different type of nitrogen than you would find in the filter paper sample? My thought would be
yes.
that the kind of nitrogens that plants use and that would be normally in animals that feed on plants and feed on uh, critters that feed on plants would be in the filter paper sample, but that the concentrated inorganic kind of nitrogen that is in fertilizers and man-made substances would be in the dissolved samples. How far off the mark am I?
I think you're simplifying a little too much. So a lot of the pollution that you're thinking of can be in that inorganic phase, but it doesn't necessarily mean that all of that is pollution. So there's always a natural component in that as well,
Which would
and it's
be things like urea, everything
yeah,
would be
yeah,
like that,
like
wouldn't
so.
it?
I mean, ammonia and nitrate, which is they're the sort of like predominant inorganic nitrogen species. They do occur naturally, and so part of the natural cycling and breakdown of nutrients eventually do sometimes break down to those species. So they can be there in natural, unpolluted systems. But yes, you're right in that some areas that their concentrations are higher because of the pollution aspect.
So what are the bad ones? Let's let
Well, the problem is like there's no bad one. It's like where it's coming from and how
yeah,
much is
yeah.
coming in.
That's right. They're
And
not
so
intrinsically bad, but
yes, yes, there's
there are
no
particular
naughty
ones.
nutrients.
There's particular ones that are causing the issues at hand. So how do you how do you determine? Is it important to know where they've come from? Like, is that
It
part of the
is.
work that you're doing?
Yeah. So, I mean, as we sort of talked about earlier, like part of the first question we're trying to answer is where are the nutrients coming from? Because you do want to know if there is this huge pollution source, maybe that's been unaccounted for, that's actually like leaking into the system, then that's something you need to address. And it would be a sort of different issue to what is actually happening. But from the work we've done so far, it isn't that there's a big influx of like nutrients from, say, a pollution source from a farm or something. The actual inflows of nutrients are low, which is what we want. It's the problem is once all these nutrients getting into the system, there hasn't been enough water flushing through the system to push out the excess nutrients. And so that's but the nutrient isotope work we've been doing is sort of led us to that outcome. So I'm sort of explaining how we've done it, but also the major finding as well.
So tell me if I'm hearing this right. I'm
Yeah.
hearing that part of the story that we've heard for many years about agricultural use in the Murray-Darling system was that we were having. Certainly, I'm of the age where salinity was this big thing that, and my first questions to you obviously showed that I haven't read enough to know that it seems to have been quite corrected. But the story has always sort of been that we're getting all these nutrients coming down, washing off agricultural land into the system. It's concentrating and it's causing problems, and the farmers need to do a better job of stopping nutrient flow. But from what I'm hearing from you is that maybe that is not the problem. That maybe the problem is, even though we probably always want to reduce what we're putting into the system,
Yes.
but that doesn't seem to be the number one problem. The number
No.
one problem seems to be that we still are not returning sufficient water to the system, however we get it, to do what needs to happen in the Kuron Lagoon.
Yes. Yeah. No, that's exactly what's happening at the moment. So it's just that buildup of nutrients, especially in the South Lagoon. Like we're just not getting enough flushing to clear those nutrients out. And so, with looking at the isotope signature of the water, the dissolved nutrients in the water, in the larger organic matter in the water, and looking at the nitrogen isotope signal in the sediment, so with the sort of like chemical processing that happens with things eating the dissolved nutrients and the breakdown of the organic matter and the deposition to the sediment, there's key changes in the isotopes that would occur. And by looking at the isotope signature of all of these different components, you can figure out what's actually occurring in the system, like with we call it nutrient cycling in the South Lagoon. So what we found was that the particulate organic matter and the sediment had a very similar nitrogen isotope signature, and that is because the organic matter that's being produced, so the algae that's growing in the South Lagoon, is then being deposited into the sediment, and that is then broken down in the sediment. But because there's so much algal material there, it uses up all the oxygen, and then you end up with this anoxic, so no oxygen present sediment. And that in turn causes that buildup of organic material in the sediment. It doesn't get broken down properly, and that leads to that like the monosulfidic black ooze, that really stinky, gross black sediment. So the fact that you're not flushing out that organic rich material in the water, it's just accumulating then over time in the sediment. And the breakdown of the nutrients then that are like the large nutrient load that's in the sediment can get re-released back into the South Lagoon. So you end up with this really unhealthy cycling of nutrients. So you've got a small amount of nutrients coming in from these freshwater flows, which aren't big enough to push out all the old water. And you've got cycling of the nutrients that are sort of like trapped in the system. So you've just got this.
So you've only ever got nutrients being added and then not being taken out.
Yes.
And because the biological system has broken down because of that, is that the concentrations just get higher and higher. Now, does that mean that insect life, fish and whatnot are really not able to survive in a natural way? Like
part of the issue is the salinity, and so the salinity is too high for their life cycle, especially in the South Lagoon. But also the lack of oxygen and everything in the sediment does stop them living there. However, some work from some colleagues has shown that, so they looked at like transplanting some of the yucky sediment into the North Lagoon to see how it would rehabilitate, and it actually started having like plants and like macroinvertebrate growing in it quite quickly, and they cleared up the sediment quite quickly. So I guess that's promising that once we get the water good, the good plants and the good bugs can come in and help rehabilitate the sediment. So there's promising stuff there as well.
I'm glad you qualified that with once we get the water good, because I'm sure there's some engineering-minded people who are going, well, why don't we just come in with some diggers and get all the crap from the South Lagoon and we'll put it in the North Lagoon, and then we don't actually have to fix the problem. We'll deal with the problem, but we won't fix it, but it won't be so bad. But that's not a solution, is it?
No. And that's, I don't think, well, I hope not being considered. But so I think the main like story that's come out of our work is that you need to get like the flushing of the lagoon right and that will facilitate rehabilitation. And in fact, the ecology itself will come in and sort of help once we get that sort of like salinity and everything down in the South Lagoon.
Okay, I want to ask you another CSI Gulwa question in a moment, but first I want to ask, I think, the hard question, which I'm not sure if you've got to that point yet.
Okay.
How much good clean water is needed to come in, and on what frequency to maintain a healthy Coorong?
So that isn't something I have looked at personally, but that's something that so through understanding what we've done and some of these other key unknowns, the government have actually put together a model. So it's a numerical simulation of the Coorong with all of this up-to-date information. And they actually run simulations looking at those kind of questions. So like how much flushing is needed to get like the water down to the end of the system. And so I think from what I looked at, there was some flows in 2016 that did flush out the system quite well. The problem is that it's not enough at the moment because the system's so degraded. So I can't give you an exact figure, but it is something that people are looking at. Like, what do we need to do to get these flows consistently
because it's
high enough?
It's a political issue, and I mean, I don't think it's a secret for anyone in Australia to understand that South Australia hasn't been happy with the way the Murray-Darling Basin Plan has operated, whereas New South Wales, Victoria and Queensland seem to think that, oh, it's all right at the moment, and it's better than it was. What are you having a whinge about? But the point is that it's still not sufficient and that more work needs to be done to return more water.
The environmental flows that we are getting are good, and they have helped improve the system, especially since the millennium drought. But I guess the South Australian government, working with the federal government, have come up with the Healthy Kuron Healthy Basin, and so that's what my work has been funded under.
Is working towards.
So we've looked at all these sort of like key knowledge gaps, and through like then providing a better understanding of the Coorong as a whole, they are now looking at possibly even some infrastructure like management options to facilitate like appropriate flushing regimes in the Coorong, so that you don't have, you still want that environmental flows from the River Murray. You still need those freshwater in flushing flows, but there might be some other, I guess, infrastructure options that can help keep flushing going all the time. And then you don't get that build up that's happened since the millennium drought.
So could that be something like directing stormwater from urban areas and whatnot into the river to supplement environmental flows, and because that water generally would eventually make its way into the system too, wouldn't it? So what kind of engineering work like that?
They haven't made a decision yet. And again, so this is my understanding of this is through being in meetings.
I know it's not your expertise.
There's multiple options that they have on the table and are looking at. So they're doing this in consultation with the community and the traditional owners to make sure that like anything that's just not appropriate isn't going to get used at the end. But I think a couple of the major options is looking at dredging in Parker Point. So as you mentioned earlier, that's shallow.
That's to keep water moving down the Coorong.
So by opening up that Parker Point area a little bit more, you facilitate water flowing from the North Lagoon, which is like healthier, into the South Lagoon, which would facilitate more fresh water coming into the South Lagoon. So that's one of the options I think that they're looking at. Another option is looking at some kind of maybe pipeline from I think it's Lake Alexandrina into maybe the South Lagoon or something like that, so that you're getting fresher water directly into the South Lagoon.
So relocating water that's already in the system. So
I guess eventually, because a lot of the water that gets flushed from the Murray Mouth does just go straight out to the ocean because it sort of mixes in that large Murray Mouth area. So I guess you're just kind of pushing it in an engineering manner down to the South Lagoon. And they're also looking at connections with the ocean to bring in, because we think of the ocean as being salty, but the South Lagoon's so salty that the ocean's less salty. So that would bring in fresh water as well in comparison to the South Lagoon. It's
infinitely more preferable than the South Lagoon. Because I think I'd heard of that where there was an idea that they would locate a pumping station at the end of the lagoon and sort of draw in salt water. So that sounds like that idea. Because the reason I asked you, Stacey,
and they're also looking at pumping out water as well. So I don't think anything's been settled on yet, but there's all these options on the table. And from the better understanding of the Coorong, they're sort of trying to figure out what would be, I guess, the best option to promote the health of the South Lagoon, especially.
And really, I asked you that question because I wanted to get some of these. We often hear about, they get discussed that there's engineering solutions. So we need to actually talk about what they actually are, so that people can get visualize what they are. Because if you're looking at water that's already in the system, there's no extra water to be had unless you reduce water loss or water use up the other end. So that means evaporation and just not using the water, allowing it to come down. Yeah. So there's only no other environmental engineering solutions up the chain actually generate more water.
And I mean, with the Murray Basin Plan, we have been given like the environmental flows, which have made an improvement. But you can't create water from nothing.
You can't magic water.
No. In Australia, we wish we could in a lot of locations.
No. But even so, if water turns up somewhere, it just means it hasn't turned up somewhere else. We're not getting new water. The point of bringing that up is that even if we're making improvements, it's still not enough, and we need to deal with. Well, that brings me to the next thing, Stacey. When I bang on about protecting the regent honeyeater and the swift parrot, people say, "But why?" So if I've got a massive cotton farm at the north end of the Murray-Darling Basin and I'm listening to us, and the message that is going through is I'm hearing people say, "Give us more water." And I say, why? Why is the health of the Coorong right down there? Who's there? Why is it important?
Well, as you said, it's the Coorong's important because it is a habitat for so many things.
It's a stronghold for water birds and fish.
Yes, migratory as well as native species that live there all the time. So it is a very significant wetland for birds in general, and so we should protect it, definitely.
Of course, I was being a shit stirrer by putting it that way, but that's the way in the end, when funding needs to be got, that's really what the question is. Who cares enough about it, and why is it important enough? So from from your point of view, you're in Adelaide. You're in the Coorong regularly doing this work. You're part of a body or part of a program that obviously views the Coorong as being important. I'd like your view on, is the promotion of the importance of the Coorong um, been enough? Like, do you think the general, you know, normie people um, really have an understanding of what the Coorong is, apart from somewhere that people go to have a fishing holiday?
That's actually a bit hard because
I know,
this is a
this
state.
is 60 minutes.
It's a state and federal funded program that we've undertaken. So I'd say that from a government perspective, that it's definitely recognised as a very significant and important wetland. And we do, as part of the project, do a lot of community engagement. And I must admit that they are extremely engaged. So the community that live around the Coorong and live from the Coorong, from the fisheries and things, are you can't be more engaged.
Absolutely,
they
super
care
energized
a lot, and they
because
know
it's
a lot
their
as
backyard.
well. Because and they've lived and worked on it for decades. So they are extremely engaged and passionate, but also a wealth of knowledge because they actually watch the system as well and have seen it over decades. But I think the general, I guess, say South Australian population, I think would still care, but they're perhaps not. I think I've been lucky enough to work and communicate with the section of the public that are super engaged and caring.
So what's the Coorong vibe in Modbury?
Yeah. I mean, I guess it depends if they've managed to have a holiday down there. Because I think once you've been to these places, your care factor, obviously, well, for starters, then you know what it is and where it is, and you obviously have a slightly higher care factor. But it's definitely an area of importance, especially to those people that live and work around it, and obviously for anyone that's environmentally minded and wants to protect these habitats would obviously, I hope, be in agreement with us.
Definitely. I mean, that's really the key, isn't it? We've got to bridge the gap to the people in Parafield and Moggle and everyone in Canamble and Gilgandra, and they all have to understand how important that the Coorong is. Apart from, they have to be able to separate the Coorong from when we talk about the health of the Murray, because they're different things.
Yes, no, that is true.
And I don't think that's really been passed out very well in any of the communications, because I think a lot of people, it's not well. I just think there's a lot of people who would rather hang on to the water than to have people fishing in the Coorong. It just. I just think that's a conflict that is inherent in the way that we use water when there's not enough of it. So.
Yeah, I mean, I think there's definitely been in the recent past a trend towards recognising the importance of water for the environment, and that's obviously come out with the environmental water allocations, and they are making a difference in the Murray-Darling Basin and also in the Coorong. So I think I'd like to think we're headed in the right direction.
No, I definitely agree, and I'm not picking a fight.
No, yeah, but it's always a negotiation, and that different people want different things for sure.
Yeah, and that's the point I always want to bring up. It's fabulous, and I'm so thankful that we're making a difference and it is getting better. But getting better is not the end of the story. We need to continually be showing that it's not enough until it is enough. And when it is enough, then we can shut up and we can all sing Kumbaya.
Well, then you need to make sure it stays enough.
That's right. Well, then it has to continue to be enough.
The one thing we haven't talked about,
we don't just want it once, do we?
Change, you know, is going to be part of the
get
story
harder
as well. So we, you know, once, like, I like to think we will get the South Lagoon to a healthy state. I think it's very promising that we will get there. And then it's a matter of keeping it there. So we don't get to this point again and have to do this whole exercise again in, you know, 10 or 20, 50 years. We keep maintaining these systems in a healthy state is key.
Stacey, how long is the project funded for? Like how long will you be working on the project?
So I have worked on the project for 2 years. So the Healthy Coorong Healthy Basin project itself is, I think it's about 6 or so years. So I think the funding was announced in, say, 2018, and they're now at the point where they're looking at like the different infrastructure options. So what it was called, the trials and scientific investigations is the government terminology. So that's what I was part of, and that was a 2-year sort of really intensive research focused part of the project. So there was like myself and others looking at nutrient dynamics, and there's also been people working on looking at like doing bird surveys and checking like where they are living, where the habitats are active. There's also been people looking at the fish species and looking at the reproductive cycles that are happening at the moment. People looking at algae and the aquatic plants. So there's been a suite of research being done, like answering some really key unknowns. So that's happened from about 2020 until now. So that's all sort of like wrapping up, and all of those reports are being submitted to the government. And I should actually say they're also available to the public if anyone's interested in seeing what we've all been up to in a huge amount of detail. So you can really dive into my isotopes if anyone's interested.
I'm sure, Stacey, you can put me in touch with some of those people, and maybe we could talk about fish and birds and algae and plant life and whatnot to sort of round out the story. So if all of your individual work in all those different sectors that you've needed to investigate and report on, if that's all wrapping up, is there going to be a really significant ongoing monitoring process as part of the project, or is that going to have to be a whole new thing?
Yeah, well, it's actually been ongoing. So the state government have been monitoring the Coorong for a number of years, and that's going to continue. And as part of this project, we've sort of advised what like additional things should be looked at as part of that monitoring. So that's definitely a part of the story. So all of this sort of feeds into the numerical modelling that the government will be doing to look at which infrastructure options they might go ahead with. And I think they will probably be happening in the next couple of years. And then like with doing that, there will be sort of further research, I think, looking at the effect of what these potential infrastructure options could be. So like if you bring in seawater, like how will that affect systems and dredging and things? So they'll be looking at like, because you don't want to do something and make it worse. We want to make sure that anything that's getting done at this point forward is an improvement. And so that's where the work is sort of focusing at the moment. But definitely monitoring is like an ongoing part of the plan, because we need to know that what's been done is working. And then, as we were just saying before, like make sure that once it is all healthy, that it stays healthy. And the only way to know that is to monitor the system. So that will definitely be ongoing, as far as I'm aware, for the next 20 plus years, hopefully.
Or off into the sunset. Now, before I come to sort of what might be next for you, Stacey. I want to go back to CSI Goolwa. How what's the what are the groovy machinery, the machines or the that you use to identify your isotopes, and I mean, do you put your white coat on and you can hold up the jar and you can go, from this is from the western slope of Mount Disappointment at Narrabri.
Yeah. Well, so the nitrogen isotopes that I was talking about, they are sort of processed in a lab. So we have some colleagues actually in Melbourne, and so they have been doing the nitrogen isotope analysis for us using a mass spectrometer.
A mass spectrometer. That's what
Yeah,
I wanted
so
you to
that is basically just this instrument that, so as I said before, different isotopes have a slightly different mass. And so this mass spectrometer takes advantage of that. And as the isotope moves through the system, they kind of like move through at different rates. And so you can measure the amount of a particular isotope relative to another one using these mass spectrometers. And it's that sort of difference in isotope ratio that we're looking at to then investigate what's happening with the nitrogen system. But what we haven't talked about, but also what I've been doing as part of my project, is looking at strontium isotopes. So this is a trace metal that's part of the lagoon. And by looking at the isotopes of the strontium, we can look at the different water sources. So we didn't touch on that so much, but that's actually what we've been doing at Adelaide Uni, using a different type of mass spectrometer.
So where does strontium come from? Because I mean, I'm a horticulturist by background, and strontium's not something that we talk about much.
No,
in the sort of trace elements, even though I understand it is a trace element. But where does it come from naturally, and what is it? What compounds is it in that are unnatural, that find its way into the water system?
Well, most of it is natural. So strontium in the water is generally sourced from the rocks that the water's in contact with.
Okay.
And so that's where it's been useful in that the ocean has a particular strontium isotopic signal because of the type of rocks the ocean water's in contact with. And the water that comes in from the River Murray has a different isotopic signal, again, because it's in contact with sort of continental rocks that have a different strontium signal. And so looking at these different strontium isotope signals, you can then look at the isotope signal in the Coorong itself and work out how much ocean water, how much River Murray water, and how much of these other sort of minor inflows are contributing to the overall water content in the Coorong. So there's a small amount of water that can come in from groundwater. That's not a very big source though. And also there's flows actually directly into the South Lagoon from the southeast through Salt Creek. So we can look at the proportion of water from all of those different sources, as well as looking at like the nutrient concentrations and isotopes.
So does the analysis that you do, whether it be for nitrogen or for strontium, it allows you to tell where the different sources have come from, like whether it's the ocean, whether it's from water flow, whether it's from runoff, as you said, from Salt Creek or something. But can you tell how long that those samples have actually been in the system? Like if they've been there hundreds of years, thousands of years, or tens of years, or 1 year? Like, are you able to do that level of analysis?
Not with these tools extremely easily, unfortunately. So the strontium in particular tells you where it's come from, but not how long it's been there for. And again, with the nitrogen isotopes, it tells you what cycling's occurring and points to different sources, but neither of those can really tell you how long the water's been there for. But I think that's where, the CSI water aspect comes into it, because you can sort of pull together other isotopes, look at how much evaporation, how much water's coming in, and try and estimate like residence times of the water in the system. So we can get to that, but it gets a little bit more complicated again, and you need different isotopes again.
Well, I've been watching Scorpion, and I just want to say, but half-life.
Well, yeah. So there's definitely different isotopes that can tell you the residence time of water, but it's easier with groundwater.
Well, I guess the reason we're having so much fun about it is that I think often we are presented with scenarios in the dramatic field that just are not real. And
I wish I could get answers as quick as it happens on TV. That would be
it's like
quite handy.
Which star did that strontium come from?
Yeah, no. I mean, it's interesting because it takes a bit longer, so it's fun to figure it out. But yeah, the answers take a lot longer to get than what you often see in movies, unfortunately.
It must be really it must be a different way of looking at the world, I think, when you can look at rocks and like a rock face at the beach or down at the 90 mile at the Great Australian Bight or something, and whereas I might be looking at the stratification layers and looking at fossils and thinking, oh wow, those angiosperms turned up here. Back then it was dominated by gymnosperms. Flighted birds came in at here, but you're looking at a whole lot of different stuff, aren't you? You're looking at all of the elements, the compounds, and your sense of time when you're looking at a rock face is probably very different to the way I would think about it.
Yeah, somewhat. I mean, I guess it depends on the water body as well. Like, so I sometimes work on groundwater systems as well, and so they have like sometimes quite long residence times. So I guess, yeah, I think just looking at the environment and having a better understanding in general just gives you a greater appreciation for timescales and like how short like our lifetimes are in general.
Stacey, usually I have to ask people some bird questions at the end, and I say, what's the best bird? And that stumps people. But as a hydrologist and a geochemist, I want to say, what's the best kind of water?
Oh, that's so hard because
I'm glad I was able to do that.
Yeah, no, you've stumped me again because I think while I've been working on the Coorong, and that's obviously a surface water body, I do think groundwater is quite mysterious, and because you can't see it, so maybe I have a slight a slight like for groundwater, slightly more, just because it really gets my CSI going, because you can't see the water body, so it makes it even more complex again, because you don't even know how much is there easily.
Rookie question though, there's different types of groundwater though, aren't there?
Yeah. So you've got depends on, I guess, the type of rock the water is in, and again you've got like same issues with like how salty the water is and how long it's been there and where it's coming from. So it's a lot of the similar questions, and actually a lot of similar traces, like the isotopic tools that you use are similar, but just not being able to see it makes the problem like a little bit more complicated again.
So when you go travelling, do you like sample water? Do you will you like I can remember the first time I went to Kuala Lumpur and got a glass of water at the hotel, and I thought, oh my god, how do people drink this stuff? Are you like that? Do you go, oh geez, and do you have a nose for water? Can you tell what's in it
No, no,
without doing?
I'd like to lie and say I've got this great palette and I can tell you where the water's from, but no, no. Unfortunately, I need slightly more sophisticated instrumentation than just my nose to tell you what's going on.
You don't have a little sampling kit that you take with you travelling and going, pH 6.4, not having any of that, thanks.
Yeah, no, but I must admit I do love a good water hole or spring or something while I'm traveling. I will definitely veer out of my way to go check out those kind of sites because I do quite enjoy going and seeing them.
So what's the best pH for drinking water?
Probably just the regular pH 7. I think
7. We want a 7.
I mean, that's kind of where it's at. I mean, and I don't know what's the trend with the alkaline water, but like once it hits your stomach with all your stomach acid, I don't think it really matters. So I steer clear of that trend.
Right. Okay, well, let me get back on track, Stacey. What's sort of your 5-year trajectory with your waterworks?
Well, it's a bit unknown at the moment. So the Kuron project itself is obviously like wrapping up. So I've written reports for the government, but as a researcher, I also have to write sort of academic peer-reviewed publications. So I'm going to be doing that for a little while, just sort of writing up some of this work a little bit more.
I actually had a look at I actually had a look at some of them.
That's good.
I was specifically very interested in uranium series dating of Great Artesian Basin, travertine deposits, implications for hydrogeology and paleoclimate. That was my favourite.
Yeah. Yeah, that's the problem. Once you start getting into isotopes, there's so many different things you can do with them. So I've worked on some very different things using my isotopic tools. But yeah, that's actually a very different.
I've got to say, I was sort of taking the piss a little bit, but some of the stuff you've been working on is really it's really important for the ongoing management of one of Australia's great assets, the Great Artesian Basin, which is very poorly understood, I think. Is that a fair thing to say?
I mean, definitely there's locations, especially like some of the deeper sections of the basin, we have very little like information about it. That's for sure. But yeah, it's such a huge resource, and we definitely need to look at it in a lot more detail. And
I think that was the point
managing of that one appropriately as well.
Well, I think that was kind of where I was going, was that it is such a massive resource, and I think that people view it a bit like it's limitless and unending, and I don't think that there's anywhere near enough information about what would be a safe level of use.
Well, the thing that it's a little bit more talked about recently is fossil water. So rather than so these like the Great Artesian Basin, especially, like it was recharged thousands, if not hundreds of thousands of years ago. So the water that we're taking out of the system actually rained, hundreds of thousands of years
old water,
like to get in there. So, and I think thinking of it as a fossil groundwater, like, other fossil resources like gas and everything. Like there's a finite reserve there that you can use. But that's another one that supports really like important habitats. So the mound springs themselves are like little homes for so many endemic species, and it's another reason to understand these systems better, is so that you make sure you don't switch off the tap to these springs, which would then like ruin these habitats as well. So water and
complex
and ecology, it's just all very, very important and intertwined, really.
Well, that's obviously why, Stacey, you were cited 143 times since 2017 in research.
Oh, that's good to know.
Yeah. 2020 was a big year for you.
Yeah, so, good.
I'm tempted to say, you know what, we should talk again about the Artesian Basin and Australian water in general. But is that sort of your thing?
Yeah, well, the hydrologist is just basically a super fancy way of saying water scientist. Like, so, and the geochemist is a fancy way of saying I like looking at like the natural variation of like chemical elements in our water system. So I'm just a water scientist.
There's another one here that just leaps out at me as being, I really want to read this. I know I won't understand it, but I really want to read groundwater salinisation and flushing during glacial interglacial cycles, insights from aquitard pore water tracer profiles in the North China Plain. Now, that sounds really cool.
Yeah,
I know I won't understand a word of it, but I'm going to give it a crack.
Oh, I'm glad. Well, if you have any questions, let me know.
One question: did you go to China for that? Because that would have been pre-pandemic if you did.
Yeah, I have been to China, and I did get to do some sampling in China, but not actually for that study, unfortunately. Like the person who wrote the article, the primary author, was the one that did most of the work. But they brought the samples to Australia, and we worked here, like on the samples. But yeah, any excuse to go and do some more sampling would be good.
Yeah. Well, fascinating thing.
Yeah. Also, the benefit of being an environmental scientist, you have to go out into the environment and take samples. So you get to go to some pretty spectacular spots like the Coorong.
Very
do your work.
Very cool. Look, Stacey, I'm so pleased that Pint of Science. Thanks, Michelle, for putting us in touch so that we could talk about the Coorong. Look, I don't know if anyone else is interested, but I would be really interested to talk about the health of Australia's water in general, if it's something you would like to, and health being one of those terribly unscientific terms that I like to throw around. But I think it's a critical thing that we need to understand more and more. Climate change is going to make us really understand how temperature, the presence of nutrients in all sorts of water bodies are going to change the way we have to use them and can use them.
Well, I think it's just it's gonna really emphasize the too much and not enough problem that we've had so far, like flash flooding in some places and droughts in others, like are just gonna be something that unfortunately we have to deal with going forward.
And of course, when we have flash flooding, we get all sorts of other nutrient loads and all sorts of other potential pollutants getting into the waterways as well. So whole suite of issues. But hopefully we can
stop it. Well, not stop it in its tracks, but at least slow it enough that we can continue to manage these systems.
Well, I'm so glad that we are able to tell a bit of a good
Yeah,
story about the Kooyong. The Kooyong.
Coorong. Yeah.
I was always going to do it again.
No, I think that's the best thing that's come out of our research. Is it's got a really promising future. Like once we get the flushing right, and the government's working very hard on that, I think that the Coorong itself will become a healthy, beautiful habitat for bird species for a long time to come. So I'm very optimistic about the future of the Coorong at this point in time.
I really want to talk to the bird guy or the bird girl,
Yes.
whoever did the bird work.
I will send emails with introductions for sure, because I'm sure that there'll probably be slightly more interest to your general audience as well. But I'm glad you had me on to talk isotopes for a little bit.
It's a nice compendium to add on, and I think the fish too, because I think when people, the general public, think about the Coorong, they think about fish and birds.
Yeah.
And I think that's sort of the starting point for the conversation in the Australian community about the Coorong. For those upriver, up the top end of the basin, good work, keep it up. You know, we just keep going. It's participation ribbon so far. You haven't got the blue ribbon yet, but it's very, 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 very positive as we go.
It's looking good.
It is. Dr. Stacey Preistley from the Healthy Coorong Project and the University of Adelaide. Actually, what's correct? University of Adelaide or Adelaide University?
I think it's officially the University of Adelaide.
There we go. I got it right.
Yeah, I think. God, hopefully I've not got it wrong. But I often flip between them.
Long as we know which august institution we're talking about. And I think we do, because the other one is University of SA, isn't it? USA.
Yeah.
Let's not. All right, let's wrap that up, Stacey. This has been the Bird Emergency. That's been the water scientist, the geochemist, the hydrologist, Dr. Stacey Preistley. I'm Grant Williams. Check us out on Twitter at Bird Emergency. Stacey, where can people check out your latest?
Actually on Twitter at Stacey Priestley as well. I think they'll be able to find me through you.
Yes, I'll link. And actually you'll be able to, I'm going to put all the stuff we've talked about today on a page which will be thebirdemergency.com/Stacey. How cool will that be?
Yeah, so I'll send you a link to the full isotope report looking at nutrients. So I'm sure everyone would love to read that.
Absolutely, and I'll of course link to the project page, which has got all the summaries and some really cool pictures. I'll put all of Stacey's cool pictures up there. And Stacey, one more ridiculous question I want to ask before you go. Are you the other Dr. Stacey? Like, did you grow up idolising Dr. Stacey from Totally Wild?
Oh, do you know what? I didn't even realise there was a Dr. Stacey from Totally Wild. I did love Totally Wild as a kid.
She kept Channel 10 afloat for about 20 years, I reckon.
Well, I must admit, I grew up in the UK originally, so I don't know if we had
you missed
Totally Wild, so I might have had the UK version.
So you missed Dr. Stacey and Agro doing the double team. It was Anne Marie and Agro, and then it was Stacey and Agro.
We might have had different ones, but I was a Totally Wild fan, like that's for sure.
Well, now I'm going to have to link to Totally Wild. Fantastic. Thanks, Stacey. Thanks, everyone.
Thank you for having me.
It's been so great talking chemicals on the Bird Emergency. Hooroo.
Thanks.